Welcome students to this video tutorial. In today's session we are going to focus on the complement system. So now, what is complement system? In simple terms, the complement system is the body's ability to enhance its immunity. So under immunity we describe to say how the adaptive and the innate immune system communicate. They also communicate through the complement system. Okay, so now in today's session we are going to focus on the following objectives, as these are going to assess our understanding, or rather what we should cover in this tutorial. We should be able to know what a complement system is. We should look at the three types of complement system. We should also outline the triggers for each complement system. Each and every complement system has what it triggers, rather to say what make it to be active. We also want to outline the cascades for the complement system, the path, how everything is done from start up to the end. And then we want to explain the functions of the complement system. With the stated objectives, let's get started. Brief history about the complement system. We should be able to know that the complement was discovered by Jules Bordet as a heat labile component of normal plasma that causes the opsonization and the killing of bacteria. So we should note some important terms, or other words. Heat labile means they can be denatured at a certain temperature. And what is that temperature? At exactly approximately 56 degrees Celsius, complement system, if at all the serum levels has been heated to that extent, about 56 degrees Celsius, the complement system gets to be inactivated. Okay, so one thing I should remind you is that the complement system is a protein, and you know that proteins get to be denatured at high temperature. For the complement system is specifically at 56 degrees Celsius. And the component of the normal plasma that causes the opsonization as well as killing of bacteria. So opsonization is more like decorating or marking the antigen for antibodies to attack it, so that it can be recognized by the body immune system. And then killing of bacteria, basically using other mechanisms on how to kill bacteria. So it is a major arm of the innate immune system. You should take note that complement is part of the innate immune system, the component. More than 30 plasma and the cell surface proteins. So it's basically more than the 30 plasma and on the cell surface proteins. We continue. So the complement system is a biochemical cascade which facilitates clearance of pathogen from the host. This one I mentioned already. So it's a biochemical cascade, meaning some steps that are undertaken for us to finally clear a pathogen from the host. The system consists of a number of small hematogenous proteins, meaning that they are found in blood, 9 factors, normally circulating as inactive zymogens. So this complement system is made up of 9 factors which circulates is in inactive form. Okay, in the body we have enzymes that are secreted inactive. Okay, they can only be activated once the target has been found. This way is also one of the body regulation of destroying itself. So with this complement system, if at all it's active, if at all it's made active, it can start destroying its own cells. Remember, the body is made up of proteins. Okay, the body is made up of proteins, so it can start destroying itself. So these particular 9 factors that we've described, they are activated by several triggers. So there are a number of triggers. There are a number of triggers to the complement system, and we're going to look at them. So I said the components are of 9. These are named in a simpler way, C1 to C9. However, there are some cleavages. In cleavage, we mean to say there are some that at some point they get to be split, okay, using different biochemical cascades and enzymes. These same factors that we've named here, C1 and C9, they get to be cleaved. And we're going to see how we cleave these things. Each component is synthesized and circulates in an active form. I emphasize why it should be synthesized and circulated in the inactive form. So now, once initial activation occurs, we are looking at the initial activation. We are going to look at the 3 pathways of the complement system by the activated different. So we are looking at the initial activation. The inactive complement component is split into fragments designated by letters. So I said these C1 to C9, they get to be cleaved upon initial activation. Okay, they get to be cleaved. So this is just more about the complement system. We want to look at the components. What are the components of the complement system? We have proenzyme. So when we say proenzyme, we mean to say we have enzymes that are actually inactive. So enzyme plasma protein cascade system, it's a enzyme plasma protein cascade system. This system is inactive. The complement has about 30 proteins. These consist of enzymes, receptors, and the complement inhibitors. Okay. So now we want to also look at the synthetic sources. Where is the complement system made? The complement system is made by the liver mainly. It's also made by the fibroblasts, macrophages, epithelial and endothelial cells. So let's know where the complement system is made. But the most important point, what's the major area of manufacture? So that's the liver. Okay. So now let's look at the function of the complement system. We have basically simple function of the complement system. The immunology questions are going to be phrased depending on what I displayed, knowing the complement system, knowing the types of complement system and what triggers each and every of these complement system, and also knowing the biochemical cascades. And then after knowing the biochemical cascades, we want you to know the functions. So let's look at the function of the complement system. The first function of the complement system is that opsonization. What is opsonization? We have a specific chemical. You'll see how we're going to cleave this chemical from the C1 to C9. We have this specific chemical, C3b. This C3b coats bacteria for phagocytosis. When you hear the word opsonization, we mean this is an antigen present. Okay, this is a foreign substance, and then the body, specifically using the complement, decorate it or put markers on it so that it can be recognized by the immune system in the body. Okay, this makes the immune system effective because in the immune system is going to recognize this foreign substance because it has been coated by C3b. We should take note of these factors in proteins. C3b is specifically for opsonization. And the complement system also causes inflammation. By now, you should have understood what the importance of inflammation. Inflammation is so very much important in removing of toxins from the body. All the mechanism that it does, chemotoxicity, chemotaxis, and all those kind of how the body removes, how the white blood cells move close to the area of infection. That is through inflammation. Okay, so we have C3A and C5A. These C3A and C5A are also known as the anaphylotoxins. What does it mean to be an anaphylotoxins? These are the ones which are responsible for inflammation reactions as well as allergic reactions. They are responsible for releasing histamine. Okay, you know histamine directly associated with the allergy. And we also have one important thing is cell lysis. When we are going to look at the biochemical cascade, we are going to look at all the complement system are going to lead at one point. And this one point is going to be mark formation. Okay, the mark formation is the process when the bacteria has been, a hole has been made in a bacteria. That's more like compromising the structure of the bacteria, meaning that you have removed the structure or the protective layer. You've removed the cell as well as cell membrane. What is this going to cause? It's going to cause water to enter into that particular bacteria. After water enters, what happens? It's going to burst. That's what we mean cell lysis. So we're going to specifically look at how things work to form mark. We also want to look at the chemotaxis. The C5A attracts neutrophils. The chemotaxis, we're looking at the fact that if at all you're having an infection, if you're having an infection somewhere, how does the body recognize, or how does the body know that that particular infection is in that particular intended area? We have the complement system that tells the body to say the infection is here. Then the C5 attracts neutrophils. Neutrophils are the white blood cells. They come to the area of infection using different methods, and then they finally reach where the infection is. That is the chemotaxis. And then the other function is that enhances the ability of the macrophages and neutrophils to phagocytize material. Also in the augmentation of the antibody response. It also enhances immunological memory, and then clearance of apoptotic cells or bodies. So basically these are the major functions of the complement system that we have looked at. Now let's have a focus on this is more like a regulation, functions that prevent damage to the host. How does the complement system? The complement system is one of the systems that is dangerous because it's a protein, and it being an enzyme, it can attack its own cells in the body. In case the complement system can't recognize its own body cells, that is what leads to a lot of autoimmune disease like Alzheimer's disease. Okay, so now I want to specifically look at how does the innate immune system, the complement system, prevent damage to the human body. It's synthesized by the liver, meaning that is the human body. And then how is it going to protect its own body? One, enhancing antigen-specific immune responses. When we say antigen-specific immune responses, we say the complement system is there. It ensures that whatever that is foreign, okay, it maintains specificity in terms of foreignness. It can only emphasize when something is foreign that the body should attack it, and not just by guesswork, or when it's too weak, it can't recognize its own cells, but it becomes so active that it sees that this is the foreign substance, this is the body cell. I believe we're together there. Maintaining homeostasis by removing immune complexes and the dead or dying cells. So now, in this particular regard, we want to remove we want to remove, we don't want to damage the host. So in case of those damaged or dead cells that have been have been found in the body, or in case of the immune system, the immune complexes have been compromised, because compromised immune complexes are going to lead to this the complement to start attacking its own cells. So all those compromised immune complexes are going to be removed, so that we only remain with the pure component of the complement that is going to prevent the body from attacking itself. Okay, so now let's look at the 3 pathways. Let's discuss 3 pathways. At this point, we are going to just name them. We have the classical pathway, we have the alternate pathway, and we have the mannose binding lectin pathway. Okay, we are going to discuss this in detail. So now, we have a classical pathway. Remember I said you should be able to know the complement, you know the types, and you know the trigger. So now we've gone to classical pathway, which is one type of the complement amongst the 3 types of complement system. We want to look at what triggers it. In other words, we are saying what activates classical pathway. This pathway is triggered by antigen-antibody complexes. Antigen-antibody complexes, we mean to say when an antigen is bound with the antibody. So what we mean here is when the foreign substances come in the body and the antibodies have bound to this antigen, meaning that they're in contact. They've come in contact. The antibodies, your defense system, have come in contact with what is foreign. Immediately they become in contact. Definitely. Okay. So these antibodies are of different types. We are saying the many triggers of these particular antibodies is we are talking about the IgG and IgM. IgG and IgM. So when antibodies bind to a pathogen, the complement protein C1 is activated. Remember I said these proteins runs from C1 to C9. We are going to look at all of them. This leads to activation of C4 and C2. Now look at this. Once protein C1 is activated, then this leads to the activation of C4 and C2. Then this C4 and C2 combines. Get me clear? Combines. Once C4 and C2, the activated C4 and C2 combines, and they are going to form what we call C3. C3 is mainly going to be talked about because it is the destination for all of the pathways. It's the destination for all of the pathways. Whatever that biochemical cascade that happens within these pathways. But at the end of the day, we need to reach C3, which is a convertase. It's an enzyme. So now, this C3 convertase C4B2A. This C4B2A is of the same form with the alternate pathway. So now, when antibodies bind to pathogen, complement protein C1 is activated. Remember, antigen-antibody complexes. This is what triggers the classical pathway. They are going to ask you questions. What triggers the classical pathway is when an antibody is bound with the pathogen. That is an antigen-antibody complex. Complex means they are bound. And where have we reached? We have reached at C3. C3 has been formed. Let's look at the alternative pathway. With the alternative pathway, what triggers the alternative pathway? The alternative pathway is triggered by microbial surfaces. Microbial surfaces. So let's get this. This is a microorganism that has come in the body, and it has been bound with the alternative pathway complement. So this does not require antibodies. Remember, the classical one needed antibodies like IgG as well as IgM. However, in alternate pathway, we don't need the antibodies. We only want microbial surfaces. So this alternate pathway involves factor B, D, and the propedin. All these factors are going to be facilitated by which is a C3A, and they are going to form what we call C3 convertase. However, this one is different because this one involves, sorry, I said C3A, it's the C3B. The C3B once combines with these factors, is going to form C3 convertase. So the complex of C3 convertase for alternative is C3BBB, and it is different from this convertase, which is the C4B and 2A, meaning the combination of C4 and the C2. However, this one is a combination of C3B and these factors. This is what makes up this complex. However, all these, whenever it's formed, is going to make what we call C3 convertase. Let's look at the lectin pathway. Lectin pathway is triggered when mannose binding lectin binds to mannose on bacteria. Binds to mannose on bacteria. So this is a trigger of lectin pathway. Mannose binding lectin binds to mannose on bacteria surfaces. Mannose are basically the carbohydrates, right? So they activate what we call an enzyme, MASP enzyme. Immediately bound, they activate MASP enzyme. These MSP enzymes lead to the activation of C4 and C2, the same activation in the classical pathway. Remember, C4 and C2 is what forms this complex, which is a C3 convertase. C4B2A. This is the complex made by C4 and C2. Same way, C4 and C2 is what makes up the C3 convertase as classical C4B2A. So now it's just the way this complex has been made. The C3 convertase appears in all of them, but the way it is made is different. In classical, it was made by antibody antigen complex, and activation of C1 led to the activation of C4 and C2. Okay, and then in the alternative, an alternative, what was in the alternative, what was the product? It was the activation of 3B. Okay, C3B. Activation of C3B combined with the factor B and D. And then these factors are what made the C3 convertase. However, with the lectin, what this mannose binding lectin, once bound on the mannose, on specifically bacteria, it activates MASP enzyme. So you can look at the activations are totally different. Okay, MASP enzyme is lectin, but we have one broad destination, that is C3 convertase. Very good. Now that you've known the trigger for each and every pathway, and you've known all the three pathways, let me try to explain a bit here. This is a classical pathway. This is an alternative pathway. This is a lectin pathway. Okay. As you can see here, this is a structure of an antibody. So antibody bound with an antigen. Okay. An antibody bound with an antigen. What forms? Activation of C1. Once C1 gets pre-activated, what happens? Let's try to write something here. So this is the classical pathway. What has happened with the classical pathway? An antibody and an antigen have made a complex. So this is going to lead to the activation of C1. C1 is going to lead to the activation of C4 and C2. C2 is going to make the C2 and C4 once combined is going to make the C3. We've seen how this C3 has been made. It has been made. Okay. How with the alternative? What has happened with the alternative pathway? With the alternative pathway, there's been a connection of this particular surface, as you can see here, with these surfaces have been connected with the microorganisms, microbial surface. Okay, microorganisms. This has led to the activation of. So let me do something different. Activation of C3b. Upon C3b activation, what happens? It combines with the protein factor B, D, so that they can form C3. Okay? And then we want to talk about the lectin. So this is a lectin, mannose binding lectin. Binds to the bacterial surface, specifically on the carbohydrates. Then what gets to be activated? It's the MSP enzyme. Once this MSP enzyme has been activated, what happens? We follow the same way. Okay, we follow the same way the the classical was activated. It starts from, sorry, not there. It starts from here. Okay, once MLSP enzyme has been activated, then the process starts from here. Then C4 and C2 gets to be activated, and what is going to lead to us is the same way. It's going to lead us again to the C3. So have we seen how these things get to be activated? Okay, now I said there are some cleavages that occurs here. Let's look at the cleavages that occurs here. What cleavages occurs here? We have reached at C3. A C3 is the destination for all of these I've explained. After C3 has been made, it's a deconvertase. So it gets to be cleaved. The first cleaving is C3A. They are just denoted by letters, the letters and numbers. Okay. So this C3 has been cleaved into C3A, which is important for the inflammation. Then C3b, which is important for opsonization, meaning a bacteria making a marker so that the neutrophils can come and destroy that particular surface. Okay? And then C3 is going to be cleaved into, it's going to be cleaved into C3b and C5b. So this C3b and C5b, they are going to combine with the C6, C7, C8, and C9. I said we're going to mention all of them. Once this combination has been made here, okay, they form what we call membrane attack complex. The membrane attack complex is MAC. Membrane attack complex, it is a hole that has been made in a bacteria so that we compromise the structure. Once the structure gets to be compromised, you understand the function of the cell membrane. It acts as a selective membrane, or rather a biological barrier. If the biological barrier has been compromised, water can easily go into the bacteria and destroy it by making it swell and burst. So the C3b, C5b, C6, C7, and C8, C9, these are simple complexes that a student should know that they form what? The MAC formation. And the MAC formation is the last step. Get me clearly? Is the last step in a complement system. Once this bacteria has been destroyed this way, then the complement system, we can say that it has done its work. Okay, so now just on to show you a step by step. Remember we said okay, this whenever you see whenever you see the Y symbol is an antibody. So the antigen and antibody have been bound. Now, guess the system? Get the pathway? What pathway is this one? Of course, antigen antibody, it's a classical pathway. They've been bound. Okay? After this has been bound, we said C1 gets to be activated. C1 gets to be activated. What happens when C1 gets to be activated? C1, once activated, it forms C2A and C4. These C2 and C4 is what we said they are fragments, okay? Specific fragments that have been made and they combine to form what we call C3 convertase. Remember, in the classical pathway, okay? In the alternative pathway, we said now this is now the continuation now. At this particular time, what happens is this. Remember we said C3 is our final destination. So for all of them, it's our midway destination for all of the classical pathway. I said the final destination is the membrane attack complex formation. Membrane, which is MAC, membrane attack complex formation. So this C3 hydrolyzes into C3b and C3a. Remember what I was showing you? The C3a is important in the inflammation. C3b is important in the opsonization. These are just fragments. And then C3b cleaves into C5. So this C3 cleaves C5 into C5a and C5b. We've formed the C5a and C5b. So whenever we add letters, we've just cleaved this particular protein. C5 has been cleaved into C5a and C5b. So now what happens is this. This C5b that has been cleaved here, that has been formed, is going to combine with the C6, C7, C8, and C9. Together they are going to form what we call a cylindrical membrane attack complex. Cylindrical membrane attack complex is a hole that is going to be formed. Because this hole has been formed, we have compromised the structure of the bacteria. Then it's going to allow water. Water is going to be formed. Upon water going inside the cell is going to swell and this is going to burst. It's a basic explanation I can give you on this side. Okay. Same here, if we look at here, okay, it's about C1 activation, the classical pathway. Then C4 and C2, they are going to combine together. They are going to be activated as well to form C3 convertase. This is going to be the destination even for the alternative pathway. Remember lectin pathway, we said they are all going to lead to C4 and C3 activation. This is one thing that you should have in mind, that the lectin and classical, they are going to lead to the C4 and C2 activation. So their C3 convertase is of the same proteins because it is made from the C4 and C2. However, the activation is different. Classical is C1. The initial activation is different. Classical is C1, and then with lectin is the mannose binding lectin on the enzyme MASP. And then with the alternative pathway, what we said that C3b is going to combine with factor B and then factor D to form this. This is different from this protein. However, the formation of this protein is the same as the C3 convertase. So we are having, at this level, we are having the same C3 convertase. Upon C3 convertase has been formed, now there is cleavage. Now we want to cleave this C3. C3 is the convertase. Here, we put it here now. This C3 is going to be cleaved into C3A. This is going to be used in inflammation. C3B is going to be used in opsonization. Okay? Then we are going to use the C3B to cleave C5. Okay? So this C3B is going to be used to cleave C5. So once C5 has been formed, it's going to be cleaved into C5A, which is also used in inflammation. Then we have C5B. This C5B is going to combine with the C6, C7, C8, C9 to form what we call membrane attack complex. Then it's going to lead to cell lysis at the end, as you can see. It is as simple as this. So this is a summary of bioactive complement peptides. Okay, these are proteins. So this is just a summary of everything that we've been talking about. I hope you liked the video and don't forget to subscribe.